This lecture is meant to be a supplement or an addendum to our previous lectures dealing with macromolecules. In our previous lectures dealing with macromolecules, we talked about a lot of different characteristics of macromolecules, including all four main categories of macromolecules. However, what I want to do is go back and touch on some concepts in both proteins as well as nucleic acids. Both of these categories of macromolecules have what's known as a polarity or a directionality. We know that these are molecules that are macromolecules and are also polymers, meaning they are linkages of one unit that is either identical or similar to one another, one right after the other. Because the units, the monomers or the monomeric units, get added on in a specific way that allows us to sort of see where the start and the end of a polymer structure is. So let's look at this particular slide dealing specifically with proteins. At this point, again, you should be very familiar with the concept that proteins are polymers of amino acids, and there are 20 distinct or different types of amino acids that can be utilized to synthesize a polypeptide. In this figure, you're looking at the addition of two amino acids, one right next to the other, with a covalent linkage that's referred to as a peptide bond. When you look at the structure of an amino acid, a single amino acid, we have the amino group, which is highlighted in the green in this slide, and the carboxyl group, which is highlighted in the pink. Specifically, the addition that occurs between the carboxyl group of the first amino acid that you see here and the amino group of the second amino acid that you see here. This again is a dehydration reaction or a condensation reaction. But if you look at the figure at the bottom, this is referred to as a dipeptide. It just means you have two amino acids that are now covalently linked to one another. More specifically, what I want to highlight with this simple dipeptide is that the amino group from the original first amino acid, highlighted in green, is still intact, whereas the carboxyl group from the second amino acid, highlighted in pink, is still intact. So even for this simple dipeptide, where the amino group is on the left-hand side, highlighted in green, is referred to as the N-terminus or the amino terminus. Conversely, on the right-hand side, you have the carboxyl group highlighted in pink. This is referred to as the C-terminus, the letter C, or the carboxyl terminus. We then went on in our previous lecture to discuss the levels of structure associated with proteins. The primary structure or the primary level associated with protein structure is what you see in this slide here. And this is again just the specific sequence of amino acids that have been linked one right after the other. In this case, you have what's known as a polypeptide. This is not truly a protein yet, and I made that clarification in the previous lecture. But what I do want to highlight on this slide is that you have the linkage of amino acids one right after the other. Up at the very top, you see the number 1, which indicates the first amino acid. And all the way at the bottom, you see the number 129, which for this polypeptide is the last amino acid. So in this example, the first amino acid, number 1, that end of the polypeptide would be referred to as the N-terminus or the amino terminus. Conversely, where amino acid number 129 is the carboxyl terminus or the C-terminus. So let's move over and talk a little bit about nucleic acids. Nucleic acids are polymers of nucleotides. There are two categories of nucleic acids, DNA and RNA. And in this figure, like we highlighted in the previous lecture, we know that the structure of a basic nucleotide contains the phosphate group, highlighted in the yellow circle, a 5-carbon sugar or a pentose sugar in the center of the molecule, in the bottom one indicated in the red, in the top figure indicated in the blue. And then lastly, you have a nitrogenous base, which just means it's a nitrogen-containing molecule that acts as a base. So that's the basic structure of a nucleic acid. Again, in the previous lecture, I highlighted that the nucleotides for DNA are different subtly from the nucleotides in RNA. In this figure, you can see in panel A the 5-carbon sugar, and specifically what I want to highlight here is the numbering that you see for the carbons in this sugar. Starting at about the 2 o'clock position, you see the number 1 with the prime designation, the little dash at the top. Then working our way down in a clockwise fashion, the 2' carbon, continuing on clockwise 3' carbon, continuing on clockwise 4' carbon, and then outside of the ring structure is the 5' carbon. So those are the carbons, the 5 carbons that make up the 5-carbon sugar. In DNA and RNA, this sugar is subtly different from one another. In DNA, the sugar is known as deoxyribose, and in RNA, the sugar is known as ribose. The only difference is at the number 2 position, the number 2 carbon position, specifically what is covalently attached to that 2' carbon position. In ribose, there is a hydroxyl group, and in deoxyribose, the oxygen is absent. But the importance of this figure here is to highlight those carbons that are part of the sugar, and again, their numbering system starting clockwise 1', 2', 3', 4', and 5'. It is this numbering system that plays a role in giving us the polarity or the directionality of a nucleic acid molecule. In this figure, what you're looking at is a polynucleotide. It is a polynucleotide because it is a linkage of nucleotides, one right after the other. More specifically, this is a single strand of RNA, and the reason we know that this is RNA is if we hone in on the sugar, we can see that this sugar is ribose and not deoxyribose. But what I want to highlight with this particular polynucleotide is up at the very top, you can see the 5' designation, and then this arrow that's superimposed over the molecular structure. The bottom of the arrow where the point is the 3' end of this polynucleotide. The way that the directionality or the polarity of a nucleic acid comes into play is whether or not the 5' carbon is linked to another nucleotide or the 3' carbon is linked to another nucleotide. So let's hone in at the top where we say that this is the 5' end of this molecule. If you look at that nucleotide at the very end, you can see the 5' carbon, part of the sugar, is bound to a phosphate group that's part of the nucleotide structure. But what you can see is there's no additional nucleotide linked to that carbon. This is the free 5' end. If we work our way all the way to the bottom of this molecule, you can see, again, focusing on the last nucleotide, specifically the sugar, the 3' carbon, while it does have a hydroxyl group that's part of the sugar structure, does not have a covalent linkage to another nucleotide. Hence, it is the free 3' end. So this molecule, again, at the top is the 5' end, at the bottom is the 3' end. And lastly, this just highlights that concept because the 5' and 3' end of the single polynucleotides are linked or listed on this slide. In DNA, we talked about the functional molecule being a double-stranded molecule, which is composed of 2 single polynucleotide DNA strands to give us the functional DNA molecule. What I didn't mention then was that in DNA, the strands are going in opposite orientation of one another. If you look very closely at any of the figures here, on the left-hand side, or the strand, the 5' end is at the top and the 3' end is at the bottom. Whereas the second single strand, the 3' end is at the top and the 5' end is at the bottom. What's really important is the previous slide in being able to recognize and identify where the 5' end and the 3' end of a polynucleotide is.